Shall
Okay.
I share my screen?
Yeah. We are live
Hmm?
on YouTube.
Hmm.
That's it, right? We're not recording the meeting. So yeah.
No. I saw a lot of registrations, Arshi and Rutuja. Do you think we should give reminders?
I think we have sent a lot of emails. I don't
हाँ,
think we
ठीक
should
है,
send. Let's see today how things go. Otherwise, we
ठीक
can
है.
do it from the next session.
हाँ,
We still have 10 minutes, so I
we
think
still
people
have
will
10
join.
minutes. Also, plant biology में there were not any slides put in, Rutuja. None of them put in slides. So obviously they are showing their own
Yeah, I
slides.
also checked.
You checked, na? हाँ, ठीक है. So Arushi, just a role distribution. YouTube live stream you've started.
Hmm.
I will do the initial slides. I will moderate in the morning. ऋतुजा आप feedback form add कर दो end में and आप वो timer you keep the timer 15 minutes for every talk. Then we'll direct them to the networking session from 1 to 2 and come back in the afternoon for the same thing. ठीक है? ऋतुजा, you might want to keep the planning document open and also the session schedule. I've also kept the
Yeah,
session schedule,
I have
kept it
kept the
open. Na?
session schedule in the
Yeah.
background.
And just remember the feedback form is the audience one, huh? Not
Yeah,
the
yeah.
mentor one and not the
Audience.
speaker one.
And
Yeah, and
entirely
the
after opening the form, that link I have to copy and paste, na?
no, see, planning document may have put the link. Can
अच्छा
you
अच्छा
check?
हाँ हाँ
Just check. Hmm. Because if you click the open form, it has to go in view mode. So I have directly put that view link in the planning document. Yeah. And then the giveaway. Giveaway also I will finalize for the first session, Arushi. So you also see how I'm. That's it. मतलब most engaging participant. You found it, Rutuja? You found it, na? Huh?
Yeah,
You
I
can't
I found it.
just
But
copy it and just to try put it in the chat window, Rutuja.
yeah,
Let me click and make sure that that's the right one.
okay.
Hmm. Huh. No, not this one, Rutuja.
Okay.
No, this is edit. It has,
Okay.
this is not from the planning document, na?
No, this is from the planning document.
No, it can't be because I
Okay,
would have put
builder
the view,
planning document you are saying.
the
Just
main
that's
planning.
it. Okay.
It's not this. This is the view. This has to be the view version, not the edit version.
I'll check. Okay, in the planning document, there is
Hmm.
PhD Cafe series Excel sheet.
Just open that.
Okay.
You've seen this document before, na, Rutuja?
Yeah, yeah.
This is the main document with the expert mentor information and all.
Okay.
So in the first sheet, just go to the bottom. General information, right? The first sheet says
Yeah.
that is all this. You've seen this document, right?
Yes, yes, I have
Yeah.
seen this, but
Now
I
go to tasks and timelines.
task.
Go down. You saw, na, the sessions are lined up. First session, huh?
Just a minute. Yeah, in this there is session schedule plant biology.
Yeah. Then do you see orange colored boxes at
Yeah,
the
yeah,
base?
yeah. Audience feedback form.
Yeah.
Okay, I'll
So
pay.
for each session, that is what is needed. Huh? For each session. Okay, Rutuja, they are different. Huh? They are not the same.
Okay.
So make sure each one is taken uniquely.
Yeah.
Hmm? So keep this document open.
Yeah, yeah. So can you confirm I have pasted it in the chat box?
Yeah, this is correct.
Okay.
Correct. Now, but remember, each session ka form is different.
Hmm.
Huh? So don't use the same form, it won't work. ठीक है.
There are people in the waiting room.
Sorry, I can't see how many people are there in the meeting room.
There are 8.
ठीक है, we should let them in. 5 minutes, huh?
Now I can let them in, no?
Arushi, shall we let them in?
I just wanted to confirm, is Rituja letting them in or should I let them in?
Either one of you.
You tell me.
Because what has happened is I think I was trying to change my name and your name got changed. So there is a confusion. I think it's just taking two people as host. And
I am co-host. It's in
yeah,
my
so it can only be one or two. I'm right now renaming yours, Karishma. I don't know. If
हाँ,
I try to change my name, I think only one person will have to do this.
ठीक है,
Yeah.
it's okay, I'll figure it out. No worries, ठीक हो जाएगा. See, today we have only 4
Rituja,
speakers.
you are able to let people in, right? You have that option.
just a minute also. Yeah,
Hmm.
yeah, I have the option to admit.
Okay, then you admit from your side.
Okay, so shall I admit them now?
Yeah.
Okay.
Actually, the Zoom background is there in IBS में ना, in case you want it.
Hi Karishma, hi Jyoti.
Hi.
Hello, Amey. Hi, Jyoti.
Hi.
Welcome. Thank you for your time. And really great to see you all. Great to see our participants also joining in, our audience rather, and also our speakers. I can see some of our speakers. So welcome everyone. And just to introduce, I'm Karishma, my colleague Arushi and Rutuja at India Bioscience. Excellent.
So it's a new initiative you have started this year, huh?
Yes, yes.
So the talk is for 20 minutes and then 5 minutes question and
10 minutes of questions.
question.
I'll go through the initial
Uh-huh.
slides.
Okay.
Yeah.
Okay.
And yes, 20 plus 10. And for plant biology, we have 4 speakers. So we have enough time for open Q&A at the end also, which can be from audience and mentors. So thank you both for coming. Thank you both. And lovely way to see plants in your background. Couldn't be more appropriate. Yeah.
Yeah, thanks to you for initiating this series, and especially with plant biology as a kickstart session.
Yes, yes, yes. Thank you. So this is supported by the company of biologists, just to give you background. And the sessions have been structured based on their journal themes.
Okay. Okay.
Okay, great to see people filtering in. We will start at little over 10 minutes, but not too far over 10 minutes. I think Yogesh, you are our first speaker. You have the honor of being the first speaker of the first ever PhD cafe. So do keep your slides ready, Yogesh. If you can hear me, do keep your slides. I see you, but do keep your slides ready.
Rituja, you're there, right? There are a few people in the waiting room. Can you just admit them?
Am I audible? Hello?
Yes, you're audible, Yogesh. Good check. Good check. You're
Yes,
audible.
Arushi.
We'll
Thank
tell
you.
you. Yeah. Just a second. All right, it's a little over 10. I think it's a good time to get started. Uh, Science in India is very excited, as you can see, Monday morning, 10 a.m. And we are here. That's a good sign. Take it as a good sign. All right, I'll slowly get started with our housekeeping instructions and our general information that we wanted to share with you about PhD Cafe. So PhD Cafe is supported by the company of biologists. It's a new initiative from India Bioscience. It was built on the premise that PhD researchers in India have limited opportunities to present their science, given visa challenges, conference registration fees, travel costs, and so on. So why not give senior to advanced PhD researchers in the country a platform to have a pre-defense, like a defense experience where they can present a full body of work to a committee, to our expert mentors, get feedback from them, and get feedback from the broader community as well. So it's like an open defense experience. The sessions have been structured based on the themes of the journal from the company of biologists, and nothing to get started. We get started right at the root, which is plant biology. You may have a few interesting puns coming along these sessions. Please bear with us. All right, we received several applications. We shortlisted them. The idea was to give advanced researchers, more senior PhD researchers a chance to talk, to give this talk. And for plant biology, we have 4 accomplished PhD researchers talking today, and they are Yogesh, Rutuja, Sushmitha, and Supriya. And in India Bioscience style, we refer to each other by first names. All right. So that sounds good. I can see some of them in the meeting room, which is a very good sign. And this is what they will be talking about. The titles of their talks are here, and we'll introduce that when each talk comes about. So please make sure all of you can hear us and see us. Maybe start with some thumbs up if you can hear us and see us. Can you? Okay, great, great. Virtual or real? Works. Excellent. Thank you. So the chat window is open and keep it active. Please keep it active to share your thoughts, engage with the session. After each talk, PhD speakers will be given 20 minutes. We will have live Q&A, the 10 minutes of live Q&A. And PhD speakers strongly encouraged to stick to 20 minutes for 2 reasons. One is we will have to stick to time. And 2, you must hear from the expert mentors. If you eat into that 10 minutes of time, you're not going to get the feedback, right? Which is a very important part of coming here. So give that 10 minutes to the expert mentors. If you see in plant biology, we also have time at the end for an open Q&A with the mentors as well as with the audience. We encourage all of us to abide by the principles where feedback should be constructive and it should be to better the work, all right? And following this 10-to-1 session, we have open networking, and our facilitator for the open networking, we'll introduce them shortly. So these are our mentors for the plant biology session. Amey from NCBS, thank you Amey for being here. Jyoti from NIPGR, thank you Jyoti for being here. And Kalika from ICET Pune, who has a conflict, might join towards the end of this session. Unfortunately, couldn't make it. But thank you to our mentors for coming on board immediately with just an email. All right, thank you so much. Our session will be followed by, the 3-hour plant biology is followed by an open networking session where we are going to discuss themes relevant to PhD researchers in India. And today's theme is postdoc careers in India. Very important theme. Postdocs in India are considered to be a threatened species, a species, exotic species. And we want Pratik, who's doing his postdoc with Vidita Vedya, to tell us, and he's an India Alliance fellow, very accomplished, to tell us what it's like to do a postdoc in India. The link is different from this meeting link. It's a no registration link that directly leads you into the session. And that is not YouTube live stream. This is YouTube live stream. So grab your lunch at your desk and join us for the networking too. Now, there's a lot of incentive for our audience to be interactive and engaging, because the most interactive audience member, the one who asks questions, gives feedback, provides support to the PhD researchers, one in every session gets a free giveaway of our e-compendium on PhD life in India, which has been put together by my colleague Ankita, who's right here in the meeting room as well. So 10 giveaways. It can't get better than this. So grab that opportunity and keep the chat window active. All right. So with that, let's get started. I will stop screen sharing. Welcome, Yogesh. Very good to see you. Yogesh, you can start screen sharing. Maybe we need to make him co-host, Rutuja and Arushi. Could we make
I
Yogesh?
don't think so. Yogesh, can you try sharing if it's necessary?
Yeah,
I'll
I
do
think
something.
it's working.
Great,
Yeah.
great. Starts here. So Yogesh is going to give us a talk on the comprehensive profiling of biochemical, metabolite, and fatty acid transition in mustard seeds and seedlings during germination and exposure to different wavelengths of light. So welcome, Yogesh. Thank you for your interest in PhD Cafe and all the best for your talk. Go ahead. So Yogesh, we will give you an indicator of an alarm clock at 20 minutes. Do start wrapping up immediately after that, okay? And we'll give 10 minutes for Q&A. Sounds good. Okay.
So good morning all. And first of all, thank you for this opportunity for me to present here in the India Biosciences PhD Cafe. So the title of my talk is Comprehensive Profiling of Biochemical, Metabolite and Fatty Acid Transitions in Mustard Seed and Seedlings during germination and exposure to different light phases. So I'll start with some stats. Like India produces about 33.2 tons of oil seeds, and that brings us to 10% of global production. We are the fourth largest oilseed producer. However, we are second largest consumer. Someone asking. Okay. Shall I continue? So however, we are the second largest consumer of oilseeds and number one importer. So there is a need. So we are not fulfilling our demand and supply of the oilseeds. So there is a need to research in the direction of oilseed production or the productivity of oils. So there is a need to understand the this seed to seedling transition, because germination is a very critical process in the establishment of seedling or in the establishment of crops that leads to their future productivity or yield. So seed to seedling transition, this is very critical process to understand, and very few studies have shown some metabolic shifts, some checkpoints in relation to the metabolome that leads to the shift from one germination stage, like the radical protrusion, to the hypocotyl elongation, to the seedling development. So there is a need to understand the metabolic profiling and the kinetic shifts during this transition. So seed is composed of several seed reserves that are stored in it for the efficient germination of seedling, and these reserves are mobilized, metabolized during this transition, and converts and develop into shoots and roots and leads to seedling. So my talk will be divided into 4 sections. First section will be the kinetic profiling of seed to seedling transition over 6 days of germination. Then, as the seeds or the seedlings are, known to have some health beneficial properties, I will be exploring their nutritional and functional properties based on certain metabolic biochemical profiles. Then, as these mustard seeds, these are known for edible oils. So I'll be explaining about the edible oil profiles of mustard oil, compare it with other mustard oil brands and other edible oil brands like coconut, groundnut, soya bean, etc. And finally, I'll be explaining when these seedlings are exposed to different light wavelengths, how their metabolism changes, how it perturbs, how different metabolic profiles are can be captured or influences through these different light wavelengths. So I'll be explaining about the effect of different light wavelengths on these seedlings. So let's first come to the seed to seedling transition. So seed to seedling transition. So we use the mustard seeds because it has very medicinal property. It is used since ages in India for culinary and medicinal properties. And it is known that the oils and other phytochemicals and minerals present in the mustard oil has various antifungal, antibacterial properties. So it's good to consider mustard for this transition, and it's a commercial crop also. So we germinated the mustard seeds and we collected the samples over different stages that correspond to different days of germination. Like from day 0 corresponds to seed, day 1 to radical emergence, radical protrusion, hypocotyl elongation. Then we shifted these hypocotyl elongated seedlings to the lights, and then alternate 16 to 8 hours light and dark period. And finally, we collected the seedlings. So we evaluated the biochemical profiles, the metabolite profiles, and fatty acid profiles. So the biochemical profiles shows that there is a counter relationship between the total lipids and the total carbohydrates. So over the initial days of germination from day 0 to day 3, these lipids are almost non-significant or similar, and the carbohydrates over total lipids, carbohydrate, and protein, these are almost similar over the day 0 to day 2. But after day 2, this total lipids are decreasing and total carbohydrates are increasing. So we had thought that uh, let's check the profile of lipids, how they are degrading. So we did the 1HNMR analysis, and we found different decrease in the levels of total lipids after significantly after day 2. That matches with our biochemical profiles. But again, here in the 1HNMR spectra, if I zoom this 2.5 ppm, and this gives to this picture where clearly the green and brown, these two peaks are separating after day 1 to day 2. So this green peak corresponds to ester bound fatty acids. So ester bound fatty acids are like all the seeds have oils in the form of triglyceride. Triglyceride means the fatty acid attached to the glycerol bond. So here the fatty acids are attached to the triglyceride in the form of triglyceride, and now after day 2 or between day 1 and day 2, these fatty acids are breaking down. So maybe the beta oxidation is majorly acting at this stage. Then we further profile uh, these seeds or the seed to seedling kinetics through metabolomics using GCMS analytical techniques, and we found 60 metabolites that corresponds to amino acid, sugar, organic acids, and various fatty acids, and certain sterols, polyphenols, and secondary metabolites. These were also identified. And then after metabolomics and metabolic profiling, we developed a simplified metabolic model for this seed to seedling kinetic transition in mustard. So let's start with the seed that has about 98% or 99% of triglycerides in oils in the lipid droplets. So these oils are over the germination after imbibition, these are degrading into fatty acids and glycerols. Fatty acids varying from myristic acid, oleic acid, linoleic, linolenic, erosic, steric. These are the general composition of fatty acid in the mustard oil, in the mustard. So then these fatty acids are going into the glyoxysomes. So glyoxysomes are lying nearby to the lipid droplets. They are degrading these fatty acids further through beta oxidation and converting into succinate. So this was confirmed by the exponential rise in the glyoxylate level in the day 0 to day 2, day 3, and then the glyoxylate levels are almost consistent. So here the beta oxidation is activating, and then these succinates after the glyoxylate cycle, these 4 carbon compounds uh, are going into the mitochondrial for further degradation and converting into malate, and these are going into the sucrose pool or the sugar pool largely. Further, after day 3, after the hypocotyl has reached and we shifted the seedlings to the light stage, the photoautotropic mode is again acting, and from here also from the CO2 assimilation, these are again adding up to the sugar pool. So these were investigated based on the kinetic profiles we mapped in through the GCMS based profiling. So after the seed to seedling kinetic transition study, we came to the seedlings and their nutritional profiling studies. So the next objective is the nutritional and metabolic profiling of selected Brassicaceae microgreens. So we selected the mustard first, and we compared that mustard with other Brassicaceae microgreens to just check that are there any differences in the Brassicaceae microgreens or within the varieties. So we selected mustard, pak choi, reddish pink, and reddish white. All these Brassicaceae microgreens shows excellent antioxidant potential based on these total polyphenols we evaluated and the DPPH assays. Further, uh, these Brassicaceae microgreens showed higher levels of both macro and micro minerals. So we did the ICP-MS analysis, and we found that most of these Brassicaceae microgreens, when consumed at 10-gram dry weight, so they can fulfill the RDA requirements from 10% to about 50% of daily RDA requirements, recommended dietary allowance based on FSSAI. So it's good to consume these brassicaceae microgreens to fulfill our microminerals need. Third, when we did the metabolic profiling, many of the phytochemicals were found, like cinebinic acid, they were higher in the brassicaceae microgreens, especially the mustard and patchouli. And lastly, when we did the fatty, because mustard is an oilseed crop, so fatty acids were evaluated, and the fatty acid profile shows they have higher oleic acid, linoleic, particularly linolenic acid. So these are essential polyunsaturated fatty acids that are good fat or good tubers and are nutritionally healthy for the human consumption. Additionally, they have some terpenes like neophytidine and some phytols. These are also health beneficial. So all these biochemical, metabolite, fatty acid profile and ionomic profile, this shows that these microgreens have health beneficial potential and can act as functional food to overcome hidden hunger. So next we profiled the mustard oil and compared with other edible oils. So we collected almost 40 edible oil from the market and profiled all the fatty acids based on 1H-NMR and the GCFID. So this 1H-NMR profile shows that, so with the 1H-NMR, we can only profile like what is the composition of what is saturated fatty acids, polyunsaturated or monounsaturated fatty acids based on these Ln, L, O and S percents. So with some formulas, we can evaluate these respective percentages. So this S corresponds to saturated fatty acids, O for MUFA or monounsaturated fatty acids, and Ln plus L corresponds to PUFA, that are polyunsaturated fatty acids. So found that mustard has a good profile of these SFA, MUFA and PUFA. And as in general, if I talk about the fatty acids, so there are some bad fats and some are good fats. So saturated fatty acids are mostly considered bad for human consumption, and monounsaturated and polyunsaturated are considered good for human consumption. So if more is the saturated fatty acids, it's harmful for consumption. So if in case of mustard, if you see this, so it has very less amount of saturated fatty acids over all the edible oils and a good amount of polyunsaturated fatty acids. So the composition of mustard oil is in some way good for human consumption. So if I compare this mustard oil profile with other oils, other popular oils like olive oil has like more of the saturated fat compared to mustard and very less of this linolenic acid, 0.65%, but mustard has around 10% of this PUFA. But again, there are certain limits and considerations while we are using these mustard oils. Like Indian cooking style is different. We use frying process. We treat the oil at higher temperature, like 180 degrees Celsius. So they can change the composition and the conformations of the edible oil and fatty acids, particularly in the edible oil. So again, based on the requirement, the need, these oils can be evaluated. Next, we further did the GC-based detailed comprehensive profiling of fatty acids, and we found that these mustard oil, they are rich in the monounsaturated fatty acid, but that is erucic acid, and erucic acid is known to be harmful for human consumption. Certain studies shows that in the rodents, when they fed with high erucic acid diet, the myocardial lesions or the deposition in their heart muscles leading to cardiovascular diseases were identified in the rodents. So so the mustard oil has high erucic acid, but recent study shows that this erucic acid can have some certain health benefits. So this is contradictory at present. So more investigations need to be done in this direction. And further, several other PUFAs, MUFAs were evaluated on over all the collected edible oils. So this comprehensive profiling of based on 1HNMR and GCFID of all the edible oils collected that are present in the Indian market, and some were collected from the abroad. So we developed finally a database. Am I audible? Hello.
Yes, yes, you are audible, Yogesh.
So finally, we developed an edible oil database after this comprehensive evaluation of fatty acids. And this edible oil database, presently it is at IIT Mandi server, but after completing it, we'll be opening it for the publics of consumers. And presently it contains 40 edible oils and both 1HNMR profiles and the GCFID based profiles. And it also has a key feature that we can design our own edible oil blends. So we developed this tool. Here, if we select any composition of different edible oils and some proportions, we can get some blend ratios. And the recommended blend ratio is, or recommended SFA MUFA PUFA ratio according to WHO or AHA associations is 1:1.5:1. So so edible oil should have a saturated fat MUFA and PUFA composition of 1:1.5:1. And no single edible oil fulfills this composition. So people are using nowadays, the research is ongoing to develop certain blends or the combination of different edible oils to get this desired ratio. So this tool can help in designing the blend ratios. So after this edible oil profiles and comparing it with other edible oils, then at last, so I did the evaluation of mustard seedlings when exposed to different light wavelengths. What is the metabolic response of those mustard seedlings? I evaluated this. So I subjected these seedlings, the 6 to 7 days old mustard seedlings, to different light wavelengths that are from 445 till far red 740 nanometer. So generally these studies are done with blue light, green light, and red light, but we also included these other wavelengths and compared with the far light. So we got the morphological differences in root and shoot length, even in the dry weight of root and shoot, and some were in deep blue and deep red light. These roots are majorly high. In green light and this far red, the shoots are higher. And again, this deep blue, red, and far. So they are giving some purplish pigmentation in the shoots. So there were morphological differences. These were through the exposure to different light wavelengths. And again, some interesting morphological variations like inward curling in the cotyledons were found in the blue light. Further, when the metabolite profiles were evaluated, we got 61 metabolic features through GCMS, and these like characterized into sugars, fatty acids, sterols, and other phytochemicals. So just for an example, when if I zoom this, sorry for the inconvenience, it was hanged. So I think it's visible now. So I'll continue. So the metabolic profile, yeah. So
Yogesh, your talk is
we,
going really well. Just to tell you, now your 10 minutes of Q&A time has started. So maybe you should wrap up a little. Sure.
I will just brief it. So we found various variations, perturbations in the profiles, like at, if I zoom this 42, 43 minute spectra, so like sugar and this enucleic acid, they are varying. And again, the essential fatty acids, linoleic and linolenic. And again, we constructed this simplified metabolic pathway for the perturbation when the seedlings were exposed to different light wavelengths. And again, these different compartments and how these are varying, like glycerine, glycine, they are contributing to the photooxidation, and again how these central metabolic pathways are influencing the fatty acid metabolism, how it is influencing, and several amino acids and like the sugars metabolism were captured. So we found that several these blue light, deep blue, and far red, these light are increasing some of the essential fatty acid composition. Like if you'll see in this figure, like linolenic acid is very high in blue light. So some of these lights or their combinations can be used in future to design some to design microgreens which have enhanced phytochemical composition. So this can be a future scope through this different wavelength studies. So finally, I will conclude with the acknowledgement. So I thank my PhD supervisor, Dr. Shyam Kumar Masakarpalli, for all the opportunity, all the advices, suggestions that has led me to complete this study. And my lab members, the School of Biosciences and Bioengineering, BioX Center, AMRC, IIT Mandi, serve and DBTs for funding. And yeah, thank you.
Thank you, Yogesh. Wonderful body of work, and very keen to see how you are making databases out of this large data you are collecting beyond publications. Well done. Well done. Okay. Really well done. Well structured talk. And I hand over to the mentors now, Jyoti and Amey. And then we have a few questions from the audience. Let's see if we can take it now or at the end of all the talks. Go ahead, Jyoti and Amey.
Yeah. Yogesh, nice talk. So your three objectives is what you have structured the talk to. As I understand, the oil part is more translational, and the other two are more you're looking at the basic science questions in the other ones.
Yeah.
So brassica, if you hear about brassica, we know that it is already very high in polyunsaturated fatty acid, and you've done a nice GC profiling also in the transition. So my question is, in this transition, now you've reported a wide variety of lipids that are involved in this seed to seedling transition. When you do GC, are you, is it a GC TOF or a normal single quad GC? Are you missing out on the unknowns, essentially, because you're capturing the very known ones, right? Linolenic acid,
Yeah.
these are highly abundant ones. So do you think if you also do a bit more advanced thing, you will get more, unknown or low abundant, I don't know, low abundant fatty acid, which might really help in that transition?
Yeah. Even my research only focused on the fatty acids.
Okay.
So I'm not capturing the sterols and other terpenes.
Mm-hmm.
So those might help me in.
So if you can do this, also try it. I mean, I don't know if you have time now, but to develop this story further, because now, for example, linolenic acid, if we take the example of linolenic acid, this seems to be a major lipid in all your in your objective 1 and also in objective 3. And this is a very abundant fatty acid, right? It is the precursor of jasmonic acid. It's very essential to the plant, and it should be in high amount. But is there any functional role known for linolenic acid in this transition from seedling to, for example, if you knock down FAD is the gene which is responsible for linolenic acid biosynthesis in brassica. So has somebody shown that if you knock out this FAD, I have seen multiple papers where they have done a functional analysis of FAD, but have they looked at the seedling transitions in this?
Okay,
Does
ma'am,
something happen to that? See, when
I
we
will look into that in
yeah,
this
you should,
direction.
because that's one thing you identified and you got a list of lipid. It will help you in your publication or future research to have a functional validation for certain lipids. Of course, we'll have a big list, but at least the major ones, if we can look at the mutants and look hard into the publication, whether they are by any chance reported. I don't know if you can do transient, knockdown or something in brassica. What I've seen here is that people make stable lines in an
Yeah.
FGR, but if you can do a transient, can you knock out at least the major gene? Because we are talking about major fatty acids. And especially for linolenic acid, it's both ways, though linolenic acid is good for health. You talked about linolenic acid in both the objectives. They are good for health, it's correct, but it also shortens the life, shelf life of the oil. So there are some groups
definitely.
which try to decrease linolenic acid
With the
as
high
a way to
temperature.
increase shelf life.
Yeah,
And
yeah.
it's so I think it's a
converts
fine
into
balance of
trans-
linolenic acid that we require. Like this, you can pick up any example of major lipids and try to look if you can, at least discuss the functional aspects of having these fatty acids. The other thing is there natural variation for polyunsaturated fatty acids in the core collection, or brassica should have a core collection of genotypes, which, the breeders would have developed. So do you have a variation in these fatty acids in this collection?
Ma'am, actually, I collected the mustard oils from the market. So I'm comparing the mustard oil that are from different brands available in the market.
No, not
Like,
of the objective to in general. I'm telling in general, there should be many mustard varieties.
So
by
definitely
the
there
breed.
will be differences in within the varieties also, because so interestingly, I got a variety named PM30 and 31 from the developed from IARI. So they are low in erucic acid, almost
Okay.
0 to 5%. So there are variation. Natural variation is there within PUFAs, MUFAs, and SFAs.
And does
Some
that
are
translate into the oils from these variety, or are there a specific lipid profile that the people need for the oil production? Can you correlate it with the?
Ma'am, there is a variation, but that variation on an average is not like rising. Like if mustard has 10 on an average 10% of SFA, so it is not going from 10 to 20 or 30. So it is near about 10, 12, 13. That's it. So in general, the SFA, MUFA, and PUFA that are SFA is near about 10%, MUFA is around 60%, and rest PUFA 20 to 30% in mustard on an average. Yeah, varieties giving difference, but that is not that significant.
And if you compare it with the last objective, so I think when you do the study systematically, it would be nice to have a collection of genotypes, which is called a core collection of each, each crop will have a core collection. That would be something that you can have for future, at least in your lab, not for you, you've done enough, but for future to have this core collection and profile the lipids in this core collection and try to see how it affects. And especially interesting is in your last objective where you're looking at you're giving UV stress. And I think that objective has a lot of potential if you take it further, because then you're trying to look what happens if you give and which lipids are increasing. And in this objective, also, I would say you should go for a more higher end lipidomics platform, because again, you are getting highly abundant lipids in here. And if you want to get the real ones, which are those, which are very less in amount, you will never get it by this GC. So I think you should go for a higher end GC to capture those variation when you give a stress. That would be my 3 main suggestions. Otherwise, it was great talk. I thought that your objectives had both a potential for translation and basic science. So all the best, Yogesh. Thank you.
Thank you. Thank you so much.
Thank you, Jyoti. And Amen, why don't you go ahead with your question?
Yeah, hi Yogesh. So similar to Jyoti, thank you very much for a very nice presentation. It was really wonderful to see enormous work that you have done, and it was a very clear talk. So Jyoti has covered most of the comments already, so I will cover something with a different perspective. Starting from the science part, I just wonder that Brassica is a very huge genus with different also polyploidy. And whatever you have covered is mainly related to Brassica napus. And I wonder whether, in general, anyone has done something related to diploid, like Brassica rapa or nigra and so on. And does it make a difference in terms of the oil profiles that you see?
So, sir, there are studies in this direction, like metabolically understanding the kinetic transitions during the germination. But those are done in the model plant Arabidopsis. That's it. And so I'm trying to understand it in a commercial crop like mustard, and again some other varieties. But most of the studies are done in Arabidopsis, and that too are till the radical protrusion, or around 48 or 72 hours of germination. So I've extended it till seedling stage and fully established seedling stage in the mustard commercial.
Right, right. So one of the point that when you mentioned this autotrophic and heterotrophic mode of seedlings, it was unclear to me, particularly when you said like in the autotrophic mode, did you take all the whole seedlings, including the cotyledonary leaves and the whole root and
Yes,
hypoprofile, or
including the
was
shoot
it
and the
also
fruit.
like right, right? So that was a total part that you took for your analysis.
Yes, yes.
Right. And one of the other thing that I was also curious to know is in terms of this edible oil database, whichever you have made, wouldn't it be having a variation depending upon the plant stage from which the oil is collected, or whichever commercial oil you have used from the market? And have you thought to make it much more finer at that? So, of course,
Yes,
this is a very brilliant effort in terms of open resource, or this database is a wonderful initiative. But something more curious in terms of how it would be used in
yes,
terms
yes.
of
So,
the
sir,
user.
right now it's in a preliminary stage. We have just profiled the oils available in the market. So it contains all the oils. They are different brands of oils. Like if I'm going for the flaxseed oil, so I have the profiles of both Patanjali and other brand. Similar is with the mustard. So I have Fortune, Dhara, First Crop. So every mustard oil that is available in the market, most of the oils right now.
Mhm.
So these are only the brands that are available in the market. I have not just included those stage specific or the variety basis. Only the oils available in the market right now, but in future we'll be including. So this is ever going database data will be added.
Right. So
Yeah.
I certainly had the two same comments as that of Jyoti. You certainly should try to knock out some of the gene directly to show a genetic evidence. And one of the other point was also in terms of the last objective where you gave different wavelengths of light. That has a tremendous potential and certainly a line to take forward, maybe not in your project, but for the lab. As a mentor, I think in terms of your presentation, it was a very great talk. I just would say that as you will be likely defending in the months to come ahead, certainly I think you should also uh, keep attention to the small things, particularly in terms of, let's say, the sample size, because in some of your graphs, I really did not find the sample size in terms of n, what is n equal to. Also, statistics play an important role. So please keep an eye on that, as well as I think you didn't really include some details, particularly in heat maps. For example, what do that color range mean? So
Oh
something
yeah.
like that, and polishing that further for your actual defense would be a really good initiative. So all the best, Yogesh, and a very wonderful talk.
Thank you, sir.
Thank you,
Thank you.
Jyoti. Thank you, Amay. And thank you, Yogesh. I mean, the first talk itself, Yogesh, excellent talk. And our mentors did their roles. Everyone played their part. Thank you. Lots of audience questions Snehal Patil has. Yogesh, what I would suggest is note these questions down. And if you
Okay.
can stay for the open Q&A, you can directly address them from the audience. It's really about keeping to time.
Okay, ma'am. Okay, yeah, sure.
Thank you, Yogesh. Thanks a lot. All
Thank
right,
you.
you can stop sharing. Our next speaker, audience and mentors is Rutuja Shirsat from Dr. Vishwanath Karad, MIT World Peace University. So, Rutuja, nice to see that you are here in the meeting room, and now you can start sharing your slides. Rutuja, you want to unmute?
Yogesh
That's
will
right.
have to stop the sharing, Karishma, before. Yogesh,
Yes,
can you please stop sharing?
Yeah. Sorry.
yeah, sure.
Also, Rituja, can you check your audio? There is a lot of echo that's happening.
Sounds good. Rutuja, we don't see your slides yet. Does anyone else see them?
No, it just says has started screen sharing. Rituja, there is some issue with your audio. I have muted you because there is a very sharp noise that is coming. Can you try rejoining with the audio? Also, we are not able to see your screen yet. Yeah, we can see your screen now. I think there is some lag
Yeah,
on her
yeah.
screen.
Let's see if this works. Rutuja, are you there? Okay. Ayushi, what I would like, we'll give it a minute more.
Yeah,
And if not, we may need to move on to the next person. Hopefully, Rutuja can fix it at her end. We can. Sushmitha, you are there, right? Sushmitha? Our next speaker,
Yeah.
Sushmitha. Yes, you're there.
Yeah.
So just keep your slides ready, Sushmitha, in case we need to. Thank you. I hope it's okay that you're presenting a few minutes
Yeah.
earlier.
Yeah.
Thank you. We'll just let you know in a minute if we need to get to you. Yeah. Thank you.
Okay.
Rutuja, is it working?
can you at least type in the chat box if you're not able to speak so that we know we are able to communicate with you?
And Supriya, I see you too. Our next speaker. Yeah, I see you too. Great. What we'll do, Arushi,
Yeah, I
for
think,
respect of everybody's time,
yeah.
Sushmitha, go ahead. Let's do your talk. We will get to Rutuja maybe if she's able to log in later. Sushmitha, thank you. Thank you for agreeing to being moved up. Yeah. Usually nobody says no to being moved up from economy to business class. We'll call it that. Uh, all right, Sushmitha. Great. All the very best. Thank you for your interest and excitement for PhD Cafe, and go ahead. All the best.
Sushmitha, can you start sharing?
Yeah. Just a second. I'm sharing. Is my slides visible? I have shared it.
They are.
Yeah.
They are. So, Sushmitha, you have 20 minutes plus 10 minutes.
Okay. Good morning, everyone. I'm Sushmitha, and my research is based on CRISPR-Cas9 multiplex editing. And today's talk will be on CRISPR-Cas9 multiplex editing of 4 SPL genes I'll be targeting in my research. That is, I'll be coming to it later. SPL9 genes for elevating grain yield-related traits in soybean. And my PI is Dr. Vinuta T, and my co-PI is Dr. Vishwanathan Chinnuswamy, sir. I'm from ICR in New Delhi. And the crop of my interest is soybean. It is, as you all know, like it is one of the world's most important seed legume in terms of high quality protein, as it contains all the essential amino acids, and it has a high value in terms of nutraceuticals, like source of isoflavones, tocopherols, etc. And it is a source of various vitamins like B3, B6, B9, and a source of various important minerals. It is having a huge global market value of 151.4 US billion dollars. So this increasing demand paves way for increasing the yields of a plant. So in soybean, plant architecture is one of the important traits to be considered in order to increase the yield of a plant. It is having a unique plant architecture, having inflorescence and pods growing at every node. And the various traits of plant which control the yield is its stem growth habit, its node number, internode length, branching pattern, leaf size and shape, which is affecting the plant photosynthetic rates. There are various strategies which could be employed for improving the plant architecture of a plant. Uh, it involves the selection of superior varieties from already available germplasm or use of various omics technologies, um, and using of gene function studies like the loss of function or the gain of function studies in order to know the function of a gene, and which could be targeted for the plant improvement, or the use of various molecular breeding strategies, NGS technologies, or the study of phenotype using the forward and the reverse genetic approaches. And one of the important recent precise robust technology is CRISPR-Cas9 editing. Here, in my research, I'm targeting 4 SPL9 genes. I mean 4 SPL9 isoforms, that is SPL9A, 9B, 9C, and 9D. These SPL9 genes are already reported as a negative regulators of auxiliary meristem formation. This auxiliary meristem formation is important as a shoot of a plant is dependent on interaction between the shoot apical meristem and the auxiliary meristems. This SPL9 genes, soybean genome has 41 SPL genes, and these SPL genes are having a conserved domain of SPB domain, that is squamous promoter binding protein domain. This is conserved domain across all the SPL9 genes. And this is a 78 amino acid residue and having a zinc finger motif and 2 zinc binding sites. It binds to a cis element of PNCG, TSEA, and coming to this structure of this gene, it is having SPL9 domain, SPL9 domain, and a two zinc finger motif and a zinc SPB domain. And in rice, it is reported as SPL14 gene is an ideal plant architecture gene. Its downregulation is improving a plant architecture. In soybean, it is the first report. And this SPL9 genes are targets for microRNA MIR156. This MIR156 SPL loop is regulating a plant transition from the juvenile to adult phase. So it is important gene which we are targeting in my research. My today's talk is divided into 3 objectives. First will be the special expression profiling of this SPL9 gene isoforms in soybean in 8 different tissues. And next will be the generation of Cas9, U6 or U3 GM-SPL9 sdRNAs multiplex construct by overlap PCR and golden gate assembly. And the third will be the transformation of soybean by using cotyledon node method of transformation. Under objective 1, first involves the characterization of SPL9 isoforms, that is SPL9A to B. This was done from phytosome database. And for my research, I have used 2 varieties, that is DS9712 and JS305 soybean varieties. First, I have checked the spatio-temporal expression pattern of these 4 genes. For this purpose, RNA was isolated from 9 different tissues like leaves, stem, root tip, shoot tip, node, internode, open flower, and unopened flower. And the real-time expression profiling of these genes was done by qRT-PCR. And for this purpose, cybergreen-based chemistry was used, and tubulin was used as a housekeeping gene. Next involved the designing of sgRNA specific for all the 4 genes. The separate guide RNAs were designed for all the 4 SPL9 genes. And for this purpose, I used Cas5finder and ChopChop databases. And after once the guide RNAs were designed, the off-target check and secondary structure analysis was performed. Later on, this multiplex sgRNA construct was obtained by using overlap PCR. For this purpose, the specific primers were prepared, and for combining all the genes, we use the overlap PCR. This was confirmed by sequencing, and later on, the multiplex cassette was transformed into E. coli DH5 alpha, and later on to agrobacterium strain AHA105, and it was screened with colon PCR, and then it was taken for transformation of soybean by agrobacterium mediated cotyledon node method. Recently, we have developed one more method, that is embryonic access. I'll be speaking later. Later of these plants, I have checked the presence of Cas9 in the bargene in order to check whether the plant is transformed or not. Later on, the guide RNA specific primers are being designed and sent for Sanger sequencing. These are the results of spatial expression profiling by QRT-PCR. This is the expression pattern of SPL9A gene and 9B, 9C, and 9D in 8 different tissues. And this, the expression pattern is more or less similar, like it is mainly expressed in shoot tip or the pod. Having said that, it is having the more role in meristematic tissues, which is the point of shoot apical meristem origination. And editing these genes is expected to lead to a more compact shoot with increased nodes and branches. Later on, for creating a multiplex sgRNA expression cassette using overlap PCR and golden gate assembly, I have used 4 different vectors, that is PYL sgRNA AT-U3D, PYL sgRNA AT-U3B, means the promoter of Arabidopsis thaliana U3D, U3B, and U61 and U629. This SPL9A was expressed under U3D promoter. SPL9B was expressed under U3B promoter. SPL9C was expressed under U61 promoter, and SPL9D was expressed under U629 promoter. This was done by overlap PCR, and later on the expression cassette of individual genes was multiplexed using golden gate assembly. This is the vector backbone which we have used. It is having the BSA1 restriction site, which we have employed for cloning into Cas9 vector, binary vector. This is the pattern, like for the overlap PCR, I have performed the PCR1 initially for combining the promoter region and the guide region of the respective 9 genes. This is the PCR1 result for SPL9A having around 150 base pair, and this is for SPL9B showing around 300 or 320 base pair, and same with the SPL9C around 340 base pair, and SPL9D around 330 base pairs. This was for combining the promoter and the guide RNA region, and this is the expression pattern of PCR2, that is for combining the guide RNA and the scaffold region. It is having the 150 base pair in all the 4 genes. Later on, the promoter, guide RNA, and scaffold regions were combined using the overlap PCR. Here, the 150 base pair, and here 150 base pair, almost it is like 300 or 264 base pair for SPL9 gene, 9A gene, and for 9B it is 350, approx 350, and here it was 150. It is around 490 base pair, or after combining the promoter, guide RNA, and the scaffold regions. Here, it was similar to 470 base pair and the 480 base pair. Later on, to enhance the efficiency of transforming into E. coli, we have made a reaction with PPS and PGS flanking primers. This will just enhance the efficacy of cloning into Cas9 binary vector. This was performed with specific PPS and PGS primers for SPL9A, 9B, 9C, and 9D genes. Later on, the here we have obtained a expression cassette of individual genes. Later on, we have multiplexed using the golden gate assembly. I have omitted some primer data here. This is the result of multiplexing after obtaining a complete 1.7 kb construct. And this is the amplification of this final multiplex construct using the PBL and PBR primers to enhance the efficacy of transformation into E. coli. Later on, this 1.7 kb fragment was confirmed by restriction and digestion using PAMH1 restriction enzyme. This is the guide RNA gene specific band, and this is the positive control. Here we can see the size fragment of 6066 base pair, and rest all fragment of 1.5 kb, 1.3 kb, and 1.2 kb. It is similar in all the fragments, but in case of the transformed colony, we have seen a 7766 base pair, which means we can say like 1.7 kb has integrated between the BSA one sides. Later on, this was confirmed with Sanger sequencing. Have depicted only SPL1, SPL9A chromatogram over here. Later on, I have taken this construct into E. coli and confirmed by colony PCR. Later on to EHA105 and confirmed with colony PCR and Sanger sequencing. And it was taken for transformation by agrobacterium-mediated cotyledon node method. This is one of the most robust protocol available till now. Here we are taking a cotyledon node. Half cotyledons were taken, and it was transformed into co-cultivation media for 3 days. Later on to shoot induction encompassing different auxins like PAP and zeatin. And here it was taken for sure after 15 days of shoot induction, it was taken for elongation. And in elongation, we were adding the GA3 and zeatin. And after the elongation, it was taken for rooting media involving 1 mg per liter of IAA. And it was taken for hardening and hardening. Later on, I have obtained 72 transformants currently. I'm in a way of screening all those plants. Now I have screened 30 of them, and among them I have received 13 positive plants. I have sent for those plants for sequencing. Here for depiction, I have taken from some 5 plants. I have checked the Cas9 and bar genes in these plants in order to confirm it is transformed. And for checking the aggregates, I have sent the samples for sequencing. It is yet to arrive. So coming for the conclusion, till date, this is the first report enabling or stacking 2 traits involving plant architecture and the yield. So the ultimate anticipated outcome of this study would be to know the to understand or to obtain the genetically edited soybean plant with high yield and optimized plant architecture, which surpass the regulatory issues concerned with GM crops. Thank you. This is the overview of my study. I have taken up 4 genes and designed the multiplex cassette transformed into Cas9 vector, and it was taken for transforming the E. coli and the agrobacterium strain, and the transformation was performed and editing was analyzed. The plant which will be obtained by this plant method is similar to conventional breeding. That is the SDN1. It will be similar to conventional breeding, and it surpassed the regulatory issues concerned with the GM crops, and it will be transient free and could be taken up for further release. Thank you. I acknowledge the lab 308 division of biochemistry and my guide Dr. Vinuta for giving me opportunity on India Bioscience for depicting my research, part of my research over here. Thank you.
Thank you, Sushmitha. Very nice graphics. The last story came together very nicely, the graphic in the last slide. Over to you, Amey and Jyoti. And Sushmitha, we may have time for some audience questions also.
Amey, go first.
Yeah, hi Sushmitha, very nice talk. And I know how difficult it is to work with soybean. So I appreciate you putting all your efforts in your graduate studies and working on this crop. It's not easy to work with soybean. I just wonder, related to the starting point, you mentioned that SPL family is pretty broad in soybean, around 41 members, if I recall it right. So I just wonder what exactly is the conservation percentage of or homology between these different isoforms that you are talking about.
Sir,
Could you just give us an overview to us about that?
Sir, actually around among the SPL9 genes, among the different isoforms, it is around 12 to 15% conserved, like unique conserved sequences are there. I mean unique sequence, sorry. 15 to 12 to 15% is unique sequences among these.
Right. So they are almost around 85%.
Yeah, 80, yeah, it is conserved.
Right. And in terms of so that explains that why you had a higher expression level in pods if we go to your first objective. So is there any database in terms of the expression search for soybean about all these 41? And what was your main aim in terms of targeting only these isoforms and not the contradictory forms, which may be a differential in different tissues that you already selected? Was there
Sir, from
any reason?
phytosome also we can know like it is already reported. Means the expression pattern of this all the SPL9 genes is reported, and also there are some literature available like where they have checked the entire soybean genome, the expression pattern of these genes. So from there we like shortlisted the SPL9 gene is involved in regulating this architecture, and they are negative regulators of yield. So, but they haven't mentioned like what isoforms or what is concerned. So we thought to target all the like 4 isoforms of SPL9 genes. This
So,
was
but
the like.
right. So have you also targeted the individual forms? And I wonder whether there is any additive effect between the different isoforms, or do they act in different combinations? Or because I think you all only have done like a complete
Yeah, multiple.
right, targeting all 4 isoforms. So
Right, right.
think so, or what are your expectations? Do you anticipate that acting additively?
Sir, anyways, I'll be checking the expression pattern of these genes after the edit in the edited plants also. That I have not. This is only in the like the control plants. So like you are right. We have to check those also. Like maybe one could be having the positive effect, one could be having the negative effect. But in my research, I'm like targeted. I have done the multiplexing and I'm going for that one only. But that plan is also there, like with the individual cassette.
Right. And one of the other point that I just wanted to confirm and was curious to know is why U3 promoter for the 2 forms and why U6 for the 2 forms? Was there any reason on that, or you wanted to test both of them?
Actually, we usually U6 is for dicots and U3 is prefer for monocots. But in our case, like we wanted to test like whether it will work or not. And those vectors were also available in the lab. So for this purpose, we use this.
Right. So in general, I think whatever numbers you mentioned for the transformation efficiency, I presume that it was more or less like 25% of the transformation efficiency.
Yeah,
What is that correct?
yeah.
Right. So I am not aware of what other methods, but I know from my lab that we are targeting tomato and generating, and wounding helps a lot in terms of increasing the transformation efficiency. And I just wonder whether you have tried something like that or would plan to do something on that lines.
Sir, actually, this is the first time we are obtaining these results in our like a lab,
Right.
because the earlier transformation efficiency, the reported transformation efficiency is around 3 to 4%.
Right, right.
Yes, of yeah. So I mean, so I have developed a new protocol having the combined embryonic axis and also the half cotyledons. So it is giving more transformants as well as the editing efficiency. So we are also like having a doubt whether it could be chimera. So like in the next generation, it could have some negative impact. So we are going for southern, and we have checked screen for these genes in using the guide specific genes in different from the different branches also. It is also giving the positive effect. More or less, we are going for southern for checking. Yeah, we are planning that.
Okay, thank you very much, Sushmitha, for all your talk and clarity, and all the best with your defense.
Thank you, sir. Thanks so much.
Hello. Hi. Hi, Sushmitha. Nice to see
Yeah.
you. IRA. I am also alumnus of IRA, so it's
Yeah.
nice to see
Right.
students from there. Can you go to your slide of proof of concept?
Yeah.
I thought that was a very, very busy slide. This is where you're starting your thesis, your presentation, and this is the most important slide in the whole presentation, which tells us why you are editing this gene. I think it can be split to 3 different slides, and you have to explain very, very clearly This is a huge family you're targeting. Why do you now take, a few of them and do editing? That's what will make you stand out from 10 others who are doing editing. You can be the first one doing editing in soybean, that doesn't matter, but your proof of concept has to be explained in a bit more clearer manner, because I didn't understand it. I understood that you have, it's the negative regulator of this meristem activity, but what are you expecting when you edit it?
Yeah, actually, we are editing a more of increased in branching. Like branching pattern is increasing in the edited plants.
Yeah,
Like,
but why this specific subgroup?
sorry,
Why this specific subgroup?
ma'am, it is reported as a negative regulator of.
Then they would have also done something else to
Yeah,
show in where did they report it? In soybean, exactly in soybean?
yeah. In soybean they have
And
reported.
they have also shown that if you mutate it, something happens to branching.
Yeah.
So then how do you stand out from your work?
I
with
mean,
what is-
ma'am, they have given the expression pattern of all the genes, and they have mentioned these are the negative regulators. And when we see in rice, this SPL14 gene, it is negative regulator, and it is having the ortholog. This SPL19 is the ortholog of SPL14 of Rhiza sativa. So there it is
you
already
believe
like,
so this ortholog would have the same function in soybean?
that is one point. And based on the expression pattern and already the available reports showing it is a negative regulator, because when they have increased the expression pattern of this MIR156, they have not directly checked the expression of means targeted the SPL19, but when they have upregulated the MIR156, they have noticed the negative phenotypic traits. So
In
based
some
on this,
other crop, right? It not in your crop of interest.
no, I mean soybean only.
Only they have shown all this
Yeah.
microRNA effect in-
MicroRNA thing they have already shown in soybean only. Based on that only, we selected this as
So maybe you can, have a separate slide and tell, because
Yeah,
unless you
have-
tell the significance, we cannot appreciate your technical part. Because your slide should be a perfect mix of why you're doing it, and why you're doing it shouldn't be a small part of your talk. Why you're doing it should be a major part of your talk, and then how you did it. Yeah, because then you are describing a technique. So it would be nice if you can have this proof of, explain a little bit what is known in soybean. These are this was what this paper showed. And now I am doing CRISPR, which will help, how will it help you advance to ask another question? So this should be very clear in your proof of concept. 2-3 slides where you explain properly, because you are talking to a broad audience. Even for us as plant biologists, it is to me also fast when you say everything in one go. And I understand that you know the topic very well, but when you present, it has to be very clear as to why you're taking this specific family. So maybe this can be split into 2-3 slides. Other than that, if it is a huge gene family, how did you design the guide RNA? If you can spend some time, and how do you know that these guide RNA are targeting, this specific class only?
Ma'am, as sir also mentioned, what is the unique and the conserved percent? We designed this guide RNA in the unique region.
Which you say is like 80% unique region.
No,
And so
not 80% is the conserved region and around 15%.
okay.
Yeah.
And
So,
then how
sorry,
many guide RNAs are there for each target?
Ma'am, single guide RNAs for each target. We have
Because
designed
with multiplexing, you are taking like of all these 4 guys.
Yeah.
And what if it doesn't cut when you take this method of single guide RNA?
Single guide RNA for all the individual
I understand,
genes.
but if you also have multiple guide RNA and one target, that's also not a bad idea. Because how do you know it will cut that single guide RNA? Even for Arabidopsis, we are using a pool of 3 guide RNAs to get good CRISPR efficiency. That's something that you can think. Of course, it's nice that you know how to make this multiplex thing, but it will not be a bad idea to pick from these 4 genes, maybe one which you think is very critical, and you can try it also in a single, that you have something to fall back on if your multiplexing strategy doesn't work.
Will concern.
Yeah.
Yeah.
And what is the phenotype that you expect in the end? What will you screen these? Of course, you have screened for the, you've screened if they are editing and all, but phenotypically, how will it be screened, these edited lines?
Ma'am, mainly the branching pattern itself is the key feature. When the branching increases, every node or a branch will have pods and inflorescence. Based on that, uh, compared to like the control plants, what is the difference in the internode length? Because in the edited plant, the internode length has to decrease and the node number has to increase. And the pod set and the inflorescence, that would be considered as a phenotypic screening.
Yeah, so if it works like that, that would be very nice because, yeah, as in you have edited all of them together. The only thing is that you should have something to fall back on if in case you know how editing in single gene itself doesn't work properly. So you should have something to fall back on your PhD that you can characterize further.
Sure, ma'am.
Otherwise, great talk, Sushmitha. I think you're doing a great work because it's very tough to do CRISPR editing in soybean. So all the best.
Thank
So,
you.
if I may add, Susmita, I just forgot to ask you about the off-target effects. You did not cover anything on that, although it was mentioned on your slide. So just keep that also as a point in mind. I know that we are running out of time, but certainly an important question for the reviewer.
वो सिंक बहुत गंदे हो रहे थे सिंक
Sorry?
मच्छर भी मर जाएंगे एक पंत दो काज
Rutuja, can you just unmute? Can you mute, please? Yeah, thank you. Go ahead, Sushmitha. Sorry about
Yeah, that's fine. Actually, I have checked for the after targets itself also, but due to this slide limit was there like 12+2, so
Right.
I omitted some of the data, but I have certainly checked for the off target.
Right. So keep that in mind as an important point for addressing the reviewer.
Sure, sir. Sure. Thank you.
Thank you, Amay. Thank you, Jyoti. Thank you, Sushmitha. Wonderful talk. And this kind of engagement is what PhD Cafe is about. So thank you for your enthusiasm. Let's circle back at the end of the session if we have more time for Q&A. Sushmitha, there's a question in the chat window for you by Kunju Lakshmi. You might want to deal with that.
Sure,
Okay.
sure.
Okay. Thank you very much. We'll circle
Thank you.
back. All right. Now, Rutuja, are you ready? Rutuja Shirsat, are you ready? Can you put your audio on, video on, and start screen sharing? Rutuja, can you hear us? Let's just maybe give it a second, Supriya. Thanks for being ready. Rutuja? Rutuja, I think you'll have to unmute. You'll have to mute her for now. Rutuja Shirsat, unfortunately, it's not yet working. Just check it again at your end. Supriya, do you mind also being promoted up half an hour?
Yes, ma'am.
Okay, go ahead. You can screen share. Rutuja,
Okay.
we'll come back to you after
Just a
this
second.
talk. Okay, go ahead. So, Amay, Jyoti, and our audience members, our next speaker is Supriya Kammar, who will be talking about the development of transgenic rice with reduced endogenous cytokinin for improved arsenic tolerance. Okay, Supriya, all the very best. You have 20
thank you, ma'am.
minutes plus 10.
Okay.
Yeah.
A very good morning to one and all. My name is Supriya S. Kammar. I'm a final year PhD student at JSS Academy of Higher Education Research, Mysuru. I'll be talking on my today's topic, that is development of transgenic rice with reduced endogenous cytokinin for improved arsenic tolerance. Starting with an introduction, arsenic, it is a toxic metalloid and classified as a class 1 carcinogen agent. It is an environmental contaminant which enters the soil and water through natural sources like volcanic eruptions and anthropogenic sources like usage of chemical-based fertilizers and arsenic contaminated water for irrigation in agricultural land. It is harmful for humans as it enters the food chain through food crop. Yes, arsenic is threat to life, as intake of arsenic causes serious problems in humans as arsenicosis and hyperpigmentation when humans are exposed for prolonged for arsenic. And also there is a negative or adverse effect seen in plant root growth and shoot development. Arsenic groundwater contamination is a serious problem worldwide, and also it is a problem in India. And mostly in northeastern part of India has higher concentration of arsenic in groundwater. Usually this groundwater is used for cultivation of food crops, which is highly contaminated with arsenic. And these food crops are high chance of getting contaminated with arsenic, making a alarming situation in India. Usually the WHO guide, as per the WHO guidelines, there is a certain threshold limit of arsenic contamination in groundwater, that is about 10 microgram per liter. But according to the report, it has been according to report in 2020, it has been increased, or it has exceeded the 10 microgram per liter of arsenic contamination. And my topic of research is in arsenic in rice. So rice is grown all over the world, as it is a staple and a primary food. And as we all know that it is a waterlogged plant, and as it is a waterlogged plant, it is having higher chance of getting contaminated both from soil as well as from water, making rice grains dangerous for human consumption. So before going to my objective, let us first understand how the arsenic will, how the plants will respond to arsenic when it encounters in soil. In soil, arsenic is present in two reduced forms, mainly arsenate and arsenite. Arsenite is a phosphate analog. As you can see here, arsenate is a structural analog of phosphate. So plant what uptake system, that is phosphate transporters, will fails to recognize phosphate, and it is being uptaken in plant cell. Once it enters the plant cell, it is arsenate is rapidly get reduced to arsenite, that is highly toxic form. So plant has some defense mechanism so that it can reduce the arsenic toxicity, so plants produces some biotheal compounds called phytochelatins. So these phytochelatins are like metal chelating compounds. These will form a complex with arsenite, and it's been sequestered or transported into plant vacuole through different transporters. So this plant, different plant has different ability of detoxification. And if it exceeds that limit of detoxification, then the plant arsenic toxicity is exhibited by the plant. So it need to be controlled. So there are two ways to control arsenic toxicity. One is to inhibit this phosphate transporter, but we cannot inhibit this phosphate transporter, as phosphate is an essential macronutrient for plant. Instead, we can increase this detoxifying machinery. And there are many studies and reports where there have been increased overexpression of phytochelatins, and also transporters have been done before, and it has shown arsenic tolerant phenotypes. Here we are interested in, and our approach is using a cytokinin, phytohormone cytokinin levels modulation to increase the arsenic detoxification in rice. But in previous study by Mohan et al., it was done in Arabidopsis thaliana, where overexpression of cytokinin gene with the constitutive promoter was done. Here you can see where it showed us arsenic tolerant phenotypes when compared to wild type in presence of arsenate. So this clearly shows that cytokinin is involved in arsenic detoxifying machinery. And also before, it was reported that cytokinin is a negative regulator of stress responsive genes. So if we lower the concentration of cytokinin, there will be increase in detoxifying machinery. But there is some limitations in this study, as in this study the constitutive promoter was used, which affected the shoot system. So based on this study, we try to develop a transgenic rice plants with a root-specific cytokinin oxidase gene, which will reduce the cytokinin in root itself and increasing the detoxifying machinery in root cells, reducing the translocation of arsenate to the shoot system, in turn reducing the accumulation of arsenic in rice grains, that is the edible part. To achieve this objective, so we develop a transgenic rice plants using agrobacterium-mediated gene transfer. We followed a Saho et al. Protocol where we induced a embryonic callus from Japonica rice variety, as it is having higher transformation ability. And it was co-cultivated with our gene of interest, that is CKX gene containing agrobacterium. And then it was selected on antibiotic media, where we got transformed callus, and that transformed callus was transferred to regeneration media. And through this process, we obtained 3 main positive transgenic lines for CKX gene as well as hygromycin resistant gene as a positive control. Further, these plant, whatever we obtained was in hemizygous condition. We need, for further analysis of transgenic plants, we need to get it in a stable stable homozygous plants. So we followed antibiotic selection and PCR confirmation for several generations, following the basic Mendel's law of segregation. We were able to obtain all 3 lines with a stable transgenic lines, where in this picture you can clearly observe that all 3 plants with showing germination or resistant to antibiotic media, that is hygromycin media, where wild type so showing susceptibility, there is no germination of wild type. So further, we went for expression analysis of relative expression analysis of CKX gene in root tissue, where we checked the we wanted to know whether there is a similar expression or difference in expression, and we found that there was a difference in expression of CKX gene, and in that event 2 or a transgenic line 2 showed us a different more expression than that of others. And also this confirmed that these 3 lines were different independent events. Along with that, we used HPT or hygromycin resistant gene as a positive control, where it showed us a more fold than of cytokinin oxidase gene, as it was having a constitutive promoter. Cytokinin is also known as a negative regulator of lateral root formation, root development. So phenotypic analysis of transgenic plants, we were expecting that there should be some visible difference in the phenotype of transgenic plants and wild type. And we found there was a significant difference in root surface and root biomass of transgenic lines with wild type plants. And also we went to analyze or quantify the cytokinin levels in transgenic lines, for which we used about 6 weeks, that is 42 days old plant, wherein which showed, according to our expectation, that there was a reduction in trans-zeatin riboside, which is a precursor of cytokinin in root. There was a huge difference or difference in cytokinin levels compared to wild type. So there was a reduction. But same results we didn't get with the trans-zeatin. Then we checked for in trans arsenic tolerance study. We found that the transgenic lines, all 3 transgenic lines showed a significant difference in root length, and also root length, roots were healthy when compared to wild type. All these plants were germinated on in arsenic supplied media of concentration 50 micromolar, and was germinated for about 10 days. There was a different difference in shoot length as well as root length. And also, interestingly, there was no difference in shoot development, that it didn't show any adverse effect on shoot development. This was like, it came it has overcome the results which was done in Arabidopsis thaliana using constitutive promoter. So this confirmed that all three positive positive independent events showed improved arsenic tolerance under concentration of 50 micromolar compared to wild type. Coming to an end, summary of my work is that usage of root-specific expression of the cytokinin oxidase gene resulted in enhanced arsenic tolerance in all three independent events, and also additionally, reduction in endogenous cytokinin increased root biomass in transgenic plants compared to wild type, and also importantly, that no adverse effect was observed in root system of transgenic plants, which overcame the usage of constitutive promoter as in previous study. So as a conclusion, we can say that reduction in cytokinin, increased detoxifying machinery in roots, which
Okay, I can see a electricity outage at Supriya's end. All right, we'll wait. We'll wait till it comes back. Yeah, Supriya, no worries. You're back.
oh, sorry, ma'am.
No
I think
worries,
there's
no
some
worries.
fluctuation in power cut. Okay. So increased root biomass, which will lead to arsenic tolerant rice phenotypes along with a mineral enriched biofortified rice. So I thank my mentor, Dr. Mohan TC and Dr. Charukeshi for mentoring me throughout this work, and also my lab group, Ajay, Subramanya, Muttama, and Meghna for supporting me. And also I thank funding agencies and JSS Academy of Higher Education and Research for providing the facility for this work. And also I thank, lastly, I thank India Bioscience for giving me this opportunity for presenting my work to the science community. Thank you one and all.
Thank you, Supriya. All right. Thanks for the presentation. And Jyoti and Amay will take it forward. And you've finished well in time, so we have enough time for question and answers.
Okay, ma'am.
Priya, excellent talk. I absolutely loved your presentation. And this idea to use CKX for arsenic tolerance, as you said, it's a huge problem in rice. And the use of this, which specific you have used one specific CKX, right? Here?
Yes, sir. Yes,
Yeah,
ma'am.
yeah. So in chickpea, it is known that if you overexpress the chickpea CKX6, this publication came from
Yes,
NIPGR
ma'am. Yes,
itself,
ma'am.
from the Bashish
Mhm.
group. We do have increased root biomass, and they have already reported it. So,
Yes, ma'am.
so in rice, there is no similar studies, right?
No,
Yeah.
ma'am.
And especially linking it to arsenic, though, definitely I don't think there are any studies. And a very conclusive data, you are also showing the hormone upregulation, which I suppose you did by LCMS, right?
LCMS, yes,
Yeah,
ma'am.
yeah. So I like the talk, and you're using a root specific promoter. You are very
Yes.
clear in your presentation. I have nothing to add. So best of luck, and I think
Thank
this
you.
is a
Thank you,
very good
ma'am.
strategy for I think do you plan to take it for field trial because it's an
Yes,
overexpression?
in future we have plans, ma'am, but we are still in arsenic assay experiments. My junior is working on it, ma'am.
So are you going to use ICP-MS for
Yes, ma'am. Yes, ma'am. For
Yeah.
quantification ICP-MS.
Yeah. So I think it will be a well-rounded story if you're able to show this all this data, and all the best.
Thank you, ma'am. Thank you.
Amay, you're mute. Yeah.
Didn't recognize that. Sorry. I second Jyoti's thought in terms of wonderful presentation with a good data and very clear. I just have a few comments, let's say.
Okay.
I wonder in terms of the root specific promoter, there are different root specific promoters also in terms of different layers of root.
Yes.
Is it throughout the root, or is it also layer specific? Because I wonder that, of course, not in your project, but down the line sometime, it would also make sense to use root specific promoters mainly to the outer layers of root. For example, like SCR or SHR and so on. So have you thought on something on that lines, whether or what kind of promoters was basically used here in your work?
In this work, we have used a mild root-specific promoter.
Mm.
And I had read one paper using a strong root-specific promoter, but it was affecting the shoot development.
Right.
So because to overcome that, like shoot development, effect on shoot development, we have used here mild root-specific. So because to overcome that, we have used it.
Right. So one of the other point was also on your slide related to this root defect and less lateral root and more of main root. I did not quite clearly understand what was that staining that you showed us.
Which slide, so?
So the orange color staining. I don't remember the slide number.
No, that was a root analysis. I did it in Vision software.
Right. So could you also, I think it would be wise to also do some kind of staining in terms of additional suberin or so on and so forth.
Okay,
Because
so
this that would clearly supplement what exactly is this phenotype resulting from, right? I think data on that part would be pretty simple for
Okay.
you presumably to generate in a relatively short time that you might have. Also in terms of the arsenic cytotoxicity, whatever you have shown, you just have shown one in terms of 50 micromolar. Was that number taken from the previous available literature?
Yes,
Or
yes, sir.
did you try a series of concentrations?
We need to study more on that, sir. First, we tried with the basic toxicity, and
Right.
then we are going for like a series of experiment using different concentration.
Right. So that is on the list.
Yes.
Okay. Anyways, very nice talk, Supriya. I don't have many
Thank
much
you.
comments, but very clear slides. So
Thank you,
appreciate it.
sir.
And
Thank you,
good
sir.
luck.
Yeah, thank you, sir.
Thank you, Supriya. Rutuja, while you check if Rutuja can join, let's take some audience questions for you, Supriya, because we have time. Rutuja, can you check if the speaker is available?
Yeah, I will update you.
Sure, thank you. And Supriya, we'll take some audience questions. So Snehal has a question. Which rice variety have you used for your arsenic toxicity study?
So I have used Japonica, Nippon berry Japonica, Oryza sativa, like variety, for as it was having higher transformation efficiency. So we used that.
I see. And she also asked a question, Nehal, also, is this variety prevalent in Northeast India, which you mentioned has a high percentage of arsenic toxicity?
No. This is a basic study. So we wanted to know how it like our gene will react to the in rice. So this is a basic study, just a basic study.
Okay. I see. All right. Then we will see any
Krishna,
other
one
questions.
quick comment.
Please.
So
Sure.
if you are going to take it to a rice variety which is usually grown in India, or you want to, after this, which variety are you planning to do transformation in?
We are still like there was
MTB 1010 is what people are usually using here, and this because you can't do it in many rice varieties in India, you're not allowed to make genetic transformations in many varieties.
Okay.
What I see in the institute is that everybody's using a variety called MTB 1010. So I don't know what happens to arsenic toxicity in this variety, but it's something that you can think.
And also we were interested in to know more about arsenic molecular mechanism behind this arsenic toxicity. So we have not still like discussed on that which variety to go for. First, we are understanding how this works in rice and whether it is going to reduce in while using root specific expression. So later we are going to.
And you're also planning to use this ABC transporter. You were telling about ABC C1.
ABC, no, ma'am. We are planning to use a strong root promoter as we are using a mild root promoter for now. Recent studies.
Thank you.
Okay.
Excellent, Supriya. Thank you, Jyoti. Any other questions for Supriya or Yogesh or Sushmita? We can open it up now, Supriya. We can open it up to all the speakers. Any other question? Remember, we do have our audience giveaway for the most engaging set of questions from the audience? Yes, there's another question. Snehal, why don't you unmute? Why don't you unmute and get a chance to ask your question? And to who is it? Is it to Yogesh, Sushmitha, or Supriya? Snehal, you can unmute.
Hi. Hi, I
Hello.
just this is question for actually.
Sorry, Snehal, we could not hear. Who is the question for? Snehal, can you hear us? The question is, will the metabolomic machinery vary that you check in other varieties? I'm just curious, but I'm not sure who the question is for. Sushmitha, Yogesh, Supriya, what guesses? Who do you think the question is for?
I'm sorry, I got disconnected.
Yeah, sure. Go ahead, Snehal.
I just wanted to congratulate Supriya on her nice talk. I think
Thank
it's
you.
a good motivation for all of us who will be presenting ahead in the future. So the question is actually meant for Supriya only.
Okay.
Actually, I got disconnected, so my chat, yeah, it's there. So will the metabolomic machinery vary that you check for in other varieties that you plan to test in the future? Uh,
So you're talking about the detoxifying mechanism, what I
yes,
explained.
yes.
So that was a general plant response to any heavy metal. So usually that mechanism is seen in most of the plant. It varies according to the plant's ability of that, not according to the varieties. Still, I have to look into that.
Great point. Okay, thank you, Supriya. Thank you. It's really amazing to see. Yes, Abdul, you have a question? Abdul? Okay. Rutuja is trying to join. Rutuja, are you able to join? Okay. I'm sure Rutuja is figuring it out how to make Rutuja join. And to add to the confusion, we have same names as well. But Rutuja, there is still a static noise. Oh, there's another question in the chat window. Namini.
Hello.
Namini, is this question for Supriya? What if this mechanism varies for different varieties of rice? Is what the question is, Supriya.
I need to look into that because as I had explained, it's a general mechanism, like the detoxifying mechanism seen in plants.
Yeah, okay, I see. I see. It's a detoxifying mechanism seen in plants. Okay. Rutuja is trying to screen share, but in the meantime, you have two expert mentors here with you who have done so much work in plant biology, so many accomplishments, all budding plant biologists, and budding is another pun. All right, budding plant biologists. Throw open questions now to Amey and Jyoti. Talk about careers in plant biology, anything else, maybe even technique specific concerns. Now, this is for everyone, including Supriya, Yogesh, and Sushmitha. Just any questions, either put it in the chat window or unmute and ask. We are waiting for questions to come in and Rutuja to join.
Ma'am, I have a question.
Yes, Yogesh. Yes, Yogesh.
So, ma'am, my background is from the metabolomics part. Okay. So is it like it will be easy for me to switch to transcriptomics or molecular to define my questions better, or like will it be I can collaborate with those who are working in the molecular biology? Hello.
Yes, Yogesh. Yes. Amey or Jyoti will take it, or both of them.
This is for your question, or for your thesis, or are you talking about in general? How do you?
In general, ma'am, in general.
So you know how postdoctoral fellowships works these days. They hire you for
Yeah.
experience, they're not going to hire you and teach you new things. Even when I'm hiring a postdoc, I try to look what does the postdoc bring to my lab that, I can use it for our research. So metabolomics is a skill that not many people have. So I think you should capitalize on it and go to a lab where they ask also, use this technique to ask a very specific fundamental question. So then in that way you can, use your expertise. You shouldn't leave it and now go and join a lab which works on developmental biology who have nothing to do with metabolomics. I think you have to build on it. For example, I have worked in the area of calcium signaling since I was a PhD student. I've just changed systems, but I've always been interested in that early signaling pathway. Um, so, you can build up on, if you really like this field, you can build up on that field and go to different labs which use these techniques, but where you can also learn something else a bit more, but capitalize on what you have. And because there is a real dearth of people with good experience in India and also abroad, very few people who are trained in this well. Also for private, I mean, it opens you a window for both. Being in academics, everybody is not going to get, get a faculty position. If you want to go to a private company, also this is going to help you in both ways. So don't leave that skill. According to me, that's what I feel.
Yeah. Thank you, ma'am. Thank you for the suggestion.
Yeah, so Yogesh, if I may add something to that one, I totally agree with Jyoti in terms of capitalizing whatever you have learned and whatever you have mastered your skills in. But having said that, I'm also of the opinion that if you get a chance to learn something new, don't miss it. And I think the way you asked the question, it seemed like you are focusing on mainly the technique oriented part. For me, I think it would be wise to start asking questions related to a biological process or a biological curiosity that you are thinking towards the next step of your career, independent to what kind of approach you might use to address that question. So certainly, I think metabolomics would be your capitalized approach, which you will propose in your next step. Thinking about the question that you are trying to answer, I would certainly encourage you to be flexible in terms of open to different techniques and different tools and so on. Certainly, one of the also important aspect is about learning languages and data analysis in today's world. And I am sure that knowing or having an experience in metabolomics, you at least know to what depth it can go. So certainly use your capitalization or the kind of experience that you have from your current role. So certainly use your capitalization or the Can you repeat it, Yogesh? I think your just voice was just cracking. Hello? Kind of
No,
experience that
I think the disturbance is from some
control.
another.
Yeah, I think it's probably. Go ahead, Yogesh. You had a counter question. You had another question.
No, that's it. That's it. Thank you. So thank you.
Thank you. Thank you. Rutuja is trying. Rutuja, we can see your screen, but we don't see the slides. So maybe you try the slides. Jyoti, Ame, there is another question from Snehal. Snehal, you want to unmute and ask? You want opinions from Dr. Ame and Dr. Vadaseri. So maybe you unmute and ask them for their opinions. Snehal?
I'm just trying to grab the opportunity when there is. I just want their opinion on whether it is necessary to continue with the PhD major topic. For example, if I'm working in plant biology or in cancer biology ahead in my postdoc, so as to just to play safe and finish my postdoc in time, or what else could be our options?
No, there's no need to play safe. I think if you get a position where you can learn something new, do that for sure. Because because in the end, if you want to start a lab, you need that diverse experience. So you don't have to stick to the same thing, but might be there. You should bring in something. You should also think from the perspective of somebody who is hiring you. So you should have that skill that you bring to that lab as well. So it's both ways, you can learn and you can also bring, because postdoc is not like a PhD student abroad, in India, anywhere. There are a lot of students even in India who's coming back from abroad, and so there is a lot of competition for a postdoctoral position. And so there definitely needs to be one selling point from your side as well, and your ability to learn new things. It's always good to learn new things.
So, Snehal, just to add to it, I think it depends on your thoughts and your interest. I'm of the opinion that it should be mainly curiosity driven. That's the first thing I consider it as a top priority. And then comes the part like whether you are fascinated to a particular institute, a particular place, a particular system, a particular topic, and etc. Certainly, I think it depends on you are the best judge to judge yourself in terms of what are your strengths, what are your weaknesses. And I would emphasize that a postdoctoral training, I would particularly also select on really improving my weaknesses to get myself trained as a next generation scientist. So I think use these as a pointers in terms of not to be afraid of getting outside your comfort zones. You know your strengths well, but I think it's a fine balance between really strengthening your strengths and then strengthening your weaknesses, which you have not covered in your PhD time.
Thank you so much. That was really insightful. Can I ask another question to both of you? Um, I just want to know, I mean, since we are still juvenile, you guys are much senior than us. What, according to you, is the expectation for a postdoc student to, like, how long should we stay in the postdoctoral research? What is the minimum? Is there a minimum threshold, or like a year or 6 months or 2 years, or is it dependent on the kind of research we are doing?
So yeah, maybe I go ahead with this one. In my view, I do not think it's like a fixed time. I just think postdoctoral research is something to get you prepared to really jump into your next career ahead. Either it be industry or either it be academia. Certainly, it's not a fixed timeline to finish a postdoctoral work. I think it depends on how well you feel comfortable to execute the next role that you are looking for. And there are different expectations depending on whether you think about academia as a career or industry as a career. In my view, academia generally requires a bit more towards your CV being shaped to fellowship, research questions, the kind of publications that you need, etc., etc. If you are thinking of an industrial role, the expectations might be much different day. So I think about this next step a bit earlier, and why are you taking that postdoctoral assignment that is literally like a bridging assignment to shape you for a long-term goal that you are thinking of. So I think it really need not have a specific timeline to do a postdoc.
I tell you
Great
now,
points.
yeah,
Yes, Jyoti, you have to add
yeah,
to this.
yeah. I've had a lot
So
of
this,
postdocs in India in my lab, thankfully. It's an exception in the Indian landscape, I think. We've had excellent postdocs since I started my lab. And one thing that I find common with these postdocs who have been in my lab is that they have been in the lab long term. They're not people who are using it as, 1 year stopgap to find another position. That is not working at all, because they neither gain in the lab. You work for 1 year, nothing happens. You need at least 2 years of work to publish anywhere. So the average time of postdocs who have done exceptionally well in my lab is 3 to 4 years. So they have been fixed on this position. They have took up a project, completed it, published it, moved forward, either become faculty or gone to another labs. but they have been very, what to say, if you have a fellowship, sometimes it's only for 2 years. Many have come on NPDF or DBTRA, so they can only have a 2-year project, 2 or 3-year projects, and in some cases, for some questions, this time is very short. So if the student is really good, the PI will find ways to, retain them for some more time, that you finish this and move forward. It's the same for abroad as well. If you go for 2 years, I doubt if you can publish anything in 2 years in any lab anywhere. So only if you are very, very serious of taking this position, you have to take, but you have to commit yourself for like at least 3 years to finish anything, decently and have a publication. You need it. Of course, as Amay said, for industry, it's completely something else. I don't even think you need a postdoc. When I was talking to the industry guys, they were like, PhD is already too much. The moment you add more degrees, it's very difficult to mold that person in a company perspective. So they are different fields, and you have to. I have had a PhD student my first PhD student. She didn't even do postdoc. After her PhD, she directly went and joined a journal as an editor. So clear-cut paths which the people have these days, and as to what they want. So think what you want and decide accordingly. Postdoc should be something that helps you, is my, publication wise, is my feel.
Makes a lot of sense. Great points. Sufficient time for a postdoc and to treat it like a bridging opportunity. But even that bridging opportunity has to produce something tangible from it for it to really count, So great points, Jyoti, Ame. Rutuja has emailed me. She's trying again. She has messaged me. She's trying again. Rutuja, let's give it another shot. Okay. This is the last over. Let's make it count.
Are you
Yeah.
able to hear me?
Yeah, we can hear you now. Finally, we can hear you. And you please try
Okay,
and
so
put your video on. Try and screen share, Rutuja. And in
yes,
the meantime,
I will
Amay and
try.
Jyoti, please go ahead, Rutuja. Namini had a question. Namini, you want to? So I'll just ask the question, Namini. Can we apply for a postdoc in any other field? For example, I am working in plant tissue culture and phytochemical studies. Can I apply in cancer studies? Is what Namini asked. Amay and Jyoti.
You can
Is
apply
my
in
screen
different.
visible, ma'am?
Yes.
So should I start?
Yeah, but we can't see your slides. Yeah, now we can see your slides, Rutuja. We can see your slides. Great.
Oh,
Can you put your video on, please?
and
And thank you all for bearing with this technical glitch. We just want to give the PhD researchers their opportunity to speak after all the work that they have done. Rutuja, we can see your screen now and we can hear you, but we don't have your video on.
are you able to see, ma'am?
Yes? Okay, I think we've lost Rutuja again. Sorry about that, everyone. Jyoti and Amay, can maybe we take Namini's question about working in another field after plant tissue culture and phytochemical studies to work in cancer studies?
Yeah, so maybe I can go ahead. I just was trying to answer it here in the chat box. So, Namini, I think certainly, yes, it is possible. There's no rule as such that you can't change field, but I think that you should be fully prepared to do so. And I'm not sure there are cases which at least I consider as rare in terms of switching fields, because I think you have to be prepared, as well as I think you should have a good reason in terms of also convince the lab that you are applying to, or the kind of industry that you are applying to, of why this would be a good switch, also on your part as well as on the person that who is going to recruit you. So I think if you have a valid reason for justifying it, certainly it's possible. And there are cases wherein people have been successful as well.
Great point, Amay. I think a valid reason. Jyoti, you want to add to that? Making such a radical switch from plant tissue culture and phytochemical studies to cancer studies?
I can tell you a good, if you are really interested in it, it will be very tough to get positions. I can give you a good example. Arnab Mukhopadhyay in NII, he did his PhD in plant biology and then now he's working on completely animal field. But it's really, where you reach, whether you get into a good lab. It's very tough, I think, in the current scenario to be hired, in a lab where you don't have much of an expertise on what you're working. It's more that.
Makes sense, Jyoti. I think that's a good point to keep in mind, Namini. Interest is one thing, but practicalities of securing the position is another thing, Great. Okay, Rutuja, we see your slides. Are you there?
Yes, ma'am, I'm here.
Okay, go
Can
ahead,
you hear
Rutuja.
me?
Yeah, now let's just give this a final shot. Please go ahead. Yeah.
Okay. And sorry for the inconvenience to everyone because of some network issue, I was not able to connect. So now I'm not able to connect to the video. So is it fine for everyone to if I give the presentation in this way?
Certainly,
But
Rutuja, go
keeping
ahead.
okay. So a very good afternoon to one and all here. So I, Ms. Rutuja Shirsat, Junior Research Fellow from the Department of Bioscience and Technology, MIT World Peace University. I'm pursuing my PhD under the guidance of Dr. Mansi Mishra and Dr. Anup Kade, who is my co-guide. So basically, I'm working on cyclotides. So the topic of my presentation here is cyclotides, nature's antimicrobial marvels with transformative potential. So the topics that I'll be covering in this presentation is we will be first seeing about the cyclotide, the peptide that I am working on, its biological activity, therapeutic application, and why we are interested in that peptide, and from where we will be where we have isolated and screened the protein. So coming to the introductory part, cyclotides are a fascinating class of plant-derived peptides which have a characteristics head to tail cyclized backbone that forms a knotted arrangement. They are not protein, but they are peptide because of their very small size. That is, they have only 28 to 37 amino acids. This structure not only contributes to their remarkable resistance to enzymatic degradation, but also enhances their potential as therapeutic agent. Researchers are actively exploring cyclotide for their potential in pharmaceutical, agriculture, application, and biotechnological innovations given their stability and bioactivity. Overall, cyclotide represent an exciting area of study in natural products and molecular biology. So, going back to the history of the cyclotide, during the 1970s, Dr. Lauren Brand visited the Congo area of the Central Africa. He noticed that the African women were drinking a herbal tea from the leaves of the plant Oldenlandia affinis, where he noticed that by taking this tea, it leads to the acceleration of the labor. This tea was called as the kalata by the local people of the Africa. Grant then found that the meaning of the kalata was fast, that is accelerating the labor. After that, 30 years later, Professor Dr. David Crick came into the limelight where he took up the work of the grant and moved forward, and where he screened many of the plant, and he was more interested to know that what type of biomolecule is present in that decoction so that it helps in the acceleration of the childbirth. So why we people are more interested in the cyclotide? First of all, cyclotide have 6 cysteine conserved residues. The 6 cysteine conserved residues are bound to each other by 3 disulfide bonds. Because of this knotted arrangement, they have cyclic backbone, and because of this cyclization, they are extremely stable to thermal and enzymatic degradation. Cyclotide, as you can see in the figure, it has 6 loops. Each loop have different amino acid sequence. Each cyclotide have 6 conserved cysteine residues, but the amino acid sequence between the loop is different. This gives each cyclotide different biological activity and therapeutic application. Cyclotides are further divided into 3 subfamilies, that is Mobius family, bracelet family, and the trypsin inhibitor. Biological activity till date shown by the cyclotide is anti-HIV, anti-cancer, antimicrobial, uterotonic. It has also been used in the drug design scaffold. Agriculturally, cyclodides have been used as an insecticide agent, nematocidal, and anti. So, coming on the biosynthesis of the cyclodides are ribosomally synthesized from precursor protein. The first study that reported the biosynthesis of cyclotide and identified them to be ribosome gene encoded product was done by the Genix. Investigation into their biosynthesis is important to both the fundamental and the applied science. Next, coming upon the biosynthesis, this schematic, we can say that cyclotides have been originated from different ancestors. Cyclotides enter the secretory pathway. Cyclotides enter the secretory pathway where folding and bond formation occurs. After folding, the precursor leaves the ER and the transcript the Golgi apparatus. Specific targeting signal is important as if absent, protein will enter the default pathway. However, cyclotide precursors are targeted to pre-vacuolar compartment and the vacuole for proteolytic processing and the cyclization. The cyclization is thought to be done by the enzyme AEP, but till date, more study is to be done on the biosynthesis of the cyclotide. So cyclotides are expressed as precursor protein. The cyclotides have been suggested to co-evolve with AEP because of its suggested role in cyclization, but this is yet to be declared. More study is going on in this prospect. Moreover, the divergent evolution of cyclotide from ancestral albumin domain was suggested based on the architecture of cyclotide precursor found in the Fabaceae plant family. However, the relationship between the precursor protein architecture and sequence and the evolutionary selection of cyclotides is still unknown. The events in the development of the circular knotted structure from the linear cyclotide precursors are not completely elucidated till date. So, my main focus of this presentation is giving you a review about the diversity of cyclotide in different plant families. So,
I think has the speaker dropped off the internet?
Yeah, I think we just lost her because
We lost her. Okay. Okay, no worries. We'll wait a few minutes, but go ahead, Rutuja.
He's in the meeting.
Yes, Rutuja.
yes, toward the diversity of the cyclotide have been found in the 5 plant families.
Rutuja, we
Ubiquitously,
can't see your
cyclotides is
screen.
found
Can
in the
others, Rutuja, just a minute. We can't see your screen. Can others see your screen?
Now her screen is on sharing.
Visible. Okay.
Okay. So cyclotides are found in the 5 plant families: Rubiaceae family, Voilaceae family, Solanaceae family, Cucurbitaceae family, and the Fabaceae family. Cyclotides are mainly and ubiquitously found in the Voilaceae and the Rubiaceae family. Cyclotides occur in a wide range of individual species. They are ubiquitously found in the Voilaceae family, having been found in every single member of that family studied so far. They occur very rarely in the Cucurbitaceae family, Solanaceae family, and the Fabaceae plant family. So I have reviewed, and our main focus is on the diversity of cyclotide in the Rubiaceae family, which is gene specific. And from this figure, you can get to know that only 7 genus from the Rubiaceae family have been screened to date or have been found to have cyclotide. So this is the review that I have done. So in the first graph, you can see the graph representing the diversity of the cyclotide in Ruby
Okay, I think Rutuja has dropped off again. Is it so? Is she in the meeting room? We really apologize for this technical issue that has been cropping up. Um, just a good point to inform you all that, yes, Arushi, go ahead.
No, I was just saying that she's not in the meeting. So
Meeting room.
probably, yeah.
A good point to let you know that we have an audience feedback form for this session. Please do click on the link and give us your feedback. Um, we look forward to that. Maybe you can, Rutuja, you want to add it again so that everyone gets it
Yeah,
updated?
I will add it in the chat box.
Sounds good. All right. I think we will proceed to wrapping up now. I think we've done our best trying to enable the final talk, but final open questions to each other, to Jyoti and Ame, to us at India Bioscience. Otherwise, you get a little time to grab your lunch and meet us at the networking session at 1 p.m.
Ma'am, are you able to hear me now?
Yeah, Rutuja, we will give this one shot, okay, Rutuja? If after this drops off, I'm sure I'm afraid we'll have to uh, call it a day, okay? Please go ahead. Yeah.
Cyclotides, so this is a graph that represents the diversity of the cyclotide in the Rubiaceae family. As you know, in the Rubiaceae family, 13,000 species are present. The number of screen species in 2008, 2013, and 2017 is mentioned here, and you can see only 10% of the plant contains cyclotides. So in the Rubiaceae family, only 10% cyclotides have been found. The next pie chart will be showing the distribution of the cyclotide in the plant screen to till date in the Rubiaceae family. So in the Rubiaceae family, there are 97% species present and only 2.4 percent of species have been screened for the presence of cyclotide. So there is an emergency and there is an it is important to screen the plants from the Rubiaceae family for the presence of cyclotide. So here is the review that approximately 13,000 species are there in the Rubiaceae family and only 26 plants contain cyclotide. The number is very less because only less number of plants have been screened. So our lab mostly work for the screening of the plants mostly from the Rubiaceae and the Violaceae family for the presence of novel cyclotides. So here we have screened plants previously not screened for cyclotide. After that, after the screening procedure, we have optimized the protocol to increase the percent yield of the protein. Then we'll be jumping on the biological activity such as anti-cancer, antibacterial, antioxidant, and the therapeutic application. So these are the references. Thank you.
Thank you, Rutuja. As you would see, Ame and Jyoti, we try to give PhD researchers at different stages. Though we prioritized advanced stages of research, we gave different stages. Rutuja seems to be early on in her work, so maybe we could, give her feedback and suggestions with that in mind.
Rituja, what is the functional relevance of cyclotides? What do you know about their role in helping plants tide over environmental stress, or what have you read about that? Why do plants have these short peptides?
Yes. Okay. These short peptides, they are categorized into the plant defense peptides. Plant defense peptides are categorized into many, such as defensins, threonins, heaven-like peptides, and one of the class of mini protein is the cyclotides. Cyclotides are present only in plants. So there is a reason why they are in plants because they are involved in the defense activity of the plant, such as they have shown some sort of insecticidal activity reported till date.
So can you elaborate on the insecticidal property? This, where you have read a paper where it does anything to the insects.
Okay, so agriculturally, as it has shown the insecticidal activity. So based on this, as it is a cyclic peptide and a cyclic protein. The cyclic, because of its cyclization, it is very stable. If we look on more of its structure, the cyclotide has a hydrophobic patch. Because of the hydrophobic patch, it has the ability to bind to the biological membrane. And because of this binding, it is able to show vast biological activity, and one of the activity is the insecticidal activity.
Which insect, to be very specific? Is it for all the insect or some kind of insect? Is it only for chewing insect? What have you read on
Are you
specific
like,
function of it?
okay,
Because this will help you also in your screening. Now you are going to find many of
yes,
this. So how you
but
can't do
basically
everything. So what will you screen for?
we first of all we will be screening the plants for the presence of cyclotide, novel cyclotides from the plant screen, not screen till date. Then we're jumping upon the anti-cancer activity. We are not as of now we have not planned from the agriculture prospect, but we are into its pharmaceutics such as anti-cancer, anti-HIV, antimicrobial.
So how will you
So
test this
answering your question
anti-cancer
is
if you go to anti-cancer, how do you plan to test?
So we have chosen some cell lines. So we have isolated the peptide, and further we have confirmed it using the HPLC and MS. And after that, now we'll be jumping upon the anti-cancer activity by uh, doing the MTT assay or the some sort of mechanism of action studies, and then we'll be moving
No,
forward
why I'm asking you
with
this so
more
much
of
is
this
how do
activity.
you, there are multiple studies in plants where people have been since years trying to find anti-cancer drugs. How do you make your study stand out? So if you have to make your study stand out, you need to choose one thing that out of it that you will study, completely with your multiple peptides that you've identified. You need to have one screening procedure that is, comprehensive, and it will answer one of these questions here, either anti-H, not do everything, because it's not possible, to get a good data if you try to do everything. So if you're taking a cancer, do that in, full detail, and don't try to do multiple other things which will dampen that story, because you have to develop it. If it
Yes,
shows anti-cancer, so what? There are hundreds of drugs that show anti-cancer activity. So you are, as a PhD student, your question is why it shows that anti-cancer activity. Can I find a, target that it, or a signaling pathway target? So you have to go deeper into one instead of
oh,
making it a very general question, especially for PhD. Yeah, other than
okay.
that, is there something which activates the production of the cyclotides? I mean, I know that one of my colleagues was working on it in plant insect interactions. Is there something which makes certain class of plants produce these kind of peptides? Or why
Uh,
is it that it's only in Fabaceae a lot?
the reason behind it is not yet opened up by the scientists, ma'am. The researchers are more working on these lines, that why this peptide is synthesized or is present in only five plant families. So the evolutionary study is in process.
Okay, okay, Rutuja. Thank you very much.
Yes.
Thanks, Jyoti. Amay, you want to take a few?
Yeah, hi Rutuja. So appreciate your efforts in terms of joining us at the end, despite of so many hurdles.
Yes.
I just felt from your presentation that you are more fascinated about the evolutionary analysis. And you mentioned also once or twice about the divergent evolution. Uh,
Yes.
so can you just briefly tell us about where do you think cyclotide and what are the divergent evolutionary aspect that you are trying to define from whatever you presented us?
Okay. So my study, uh, here I'm presenting the diversity related part, but my main study is basically on the screening of the novel plants. So talking about the evolutionary divergence, sir, I need to read more about it because my main study is not mainly focused on that. I have read some of the papers because I was fascinated that how this peptide came into the limelight, or how the evolution, what is the evolutionary part of XXS. But more studies are in hand, and there are some questions in the research paper itself that how the evolution have occurred of the cyclotide.
Right. So moving a little bit beyond that, I think it's also really fascinating to catch this evolutionary investigation.
Okay.
To ask question like if they are present in only very specific plant families, are these plants completely resistant to bacteria? Not, right? So
No,
for
sir.
example, Solenaceae, it has some cyclotide peptides, but all of these plant families do have a handful of bacterial diseases. So why is that, and what exactly leads to activation of such kind of peptides? Secondly, one could also think about like where did they came into these plant families from, right? So which was the first basis of origin of these cyclotides? As far as I can recall, I don't know whether I'm correct or not, but some soil bacteria also do have cyclotides. So could that lead to the hypothesis that whether these kind of molecules came by horizontal gene transfer from bacteria to plants in the first instance, which were beneficial in terms of helping the plants to evade land much more, right? So I think talking about the fascination that you have for evolutionary work, I think you should be open to asking such kind of questions, which would be more defined rather than very general.
Okay. Okay, sir. So it is a very new thing for me to hear that cyclotides have been found in the bacteria also. So I think I should more focus on this so that I can get an evolutionary point from where this have been evolutionized.
Right. So I think, as I said, I mean, I can just recall from whatever I have read previously, but you should confirm about this. And I
Yes.
think it would be a very nice investigation in terms of the evolutionary analysis of the molecules that you are interested in.
Okay, okay. Sure, sir, sure.
Wonderful. Rutuja, happy we made it happen. Thank you, Amay, Jyoti. Thank you to all our audience members who have stayed on. You know, that shows their commitment. We have a feedback form, but I think it's time to wrap up. So, Rutuja, now I will share slides if that's okay. Yeah. So thank you very much, Jyoti and Amay. Thank you so much for your time and detailed feedback and curating it as per each one's talk and each one's stage in their career. We really appreciate it. You've set the bar very high for mentors.
Thank you.
Thank you. Thank you for
Thank you.
that.
And our speakers have set the bar very high for PhD speakers. So great conversations. It was also very nice to see them engaging with each other in the chat window. And that is a sort of community that, we want to build with this. So everyone, you have some time to eat lunch. Grab lunch and bring it to your desk or figure it out. We have an open networking session from 1 to 2 p.m. Prateek will join us, who will talk about India Alliance fellowships, being a postdoc in India, opportunities and challenges. So postdocing in India, right? Very relevant. This is the Zoom meeting link. This is the QR code. Click when you want to enter the meeting room because there's no registration link. Okay, it's going to directly lead you into the meeting room. We look forward to seeing you. Drop by anytime between 1 and 2 p.m. We will be there. And at 2 p.m., we will go into our next session. So we've done the open Q&A. We've given you the feedback form. Let us know. And we will see you at the next session today afternoon, which is cellular biology. The expert mentors are Deepa Subramaniam and Sandhya Kaushika. And we have a packed session. We have 6 speakers and we really need to stick to time. So there's going to be lots of exciting stuff happening in the afternoon as well. So drop by from 1 to 2 p.m. Otherwise, see you in the afternoon. So thank you very much, all of you again. Thank you, Team India Bioscience, and see you soon.
Thank you.
Take care. Bye-bye. Bye.